I'm John Osha, the founder and president of CompuDopt, and we're very, very excited to have all of you and also Dr. Miller at our first Technology for Tomorrow breakfast lecture. And this, we think, segues so nicely with CompuDopt's mission. And as most of you know, CompuDopt exists to provide computers and computer technology education to children who otherwise would not have access to it. And we assume that children, like our children, have a computer. They have computers in school. They have access to these things. And in fact, in many places around Houston, they don't. We've actually distributed computers to groups of middle school children who didn't know how to use a mouse. They had literally not touched a computer. And this isn't a third world country. This is Houston. So it's really amazing. And we're very, very excited that in March of this year, we will be donating our 4,000th computer to a Houston area child. So it's pretty exciting in 7 years that we've gotten that far. In addition to the computer donation part, which is a big part of the operation, we're very keyed in on the education component, because without the education component, having the hardware there doesn't do very much. So we have two other programs. One is an after-school program where we invite these children to come back and to learn more about the specifics of how to use a computer. And we have some fun programs like how to design a video game, how to do a YouTube video, things like that the kids want to come do. And of course, they don't realize they're learning along the way. And also, we have what we call a Learn to Earn program for high school kids, where they come into our lab and they learn how to work on the computers that were given away to the other kids. So they learn how to take apart a computer and service it and stuff like that. And at the end of a certain number of hours, they get to build their own laptop and take it home. So they can go through our parts bin and pull out all the best parts and build themselves a cool machine that they're happy with, and they get to keep that.
We were going to have one of our Learn to Earn students come here and introduce Dr. Miller. We were going to have about 7 students here this morning, so you can meet them and talk with them about our programs. Unfortunately, of course, because of the weather and school closings, they weren't able to make it. The students did spend a lot of time practicing the script to introduce Dr. Miller, so to honor the students that we serve and exist to serve, I'd like to read you the script that they prepared that they were going to use to introduce Dr. Miller. I'm currently enrolled in CompuDopt's Learn to Earn program, and I'm learning how to refurbish computers. Once I complete the program, I'll earn my own laptop. Aside from giving me the ability to complete my schoolwork and research my future career, these laptops support technologies such as those invented by Dr. Miller. A pioneer in tissue engineering research, he uses 3D printing to print temporary templates of blood vessels made from sugar. Dr. Miller is known worldwide for his work. He's an assistant professor in the Department of Bioengineering at Rice University. Was recently named a core developer of the RepRap 3D printer project and is the recipient of numerous prestigious fellowships. With these high-tech innovations and the opportunity to earn my own laptop, I'd like to think that I can be the next Dr. Miller. Thank you for being here this morning, Dr. Miller.
So I think the CompuDopt story is actually very similar to the story that you hear about today, which is about using 3D printers. And our story actually started in our local hackerspace because we didn't know how we were going to be able to do this medical research at the time. And so we went to our local hackerspaces, people that are sharing a workspace, sharing equipment that people are throwing away or people are donating. So a lot of the old computers that we had available, we were able to do some really interesting science with. So with that, I'll just kind of start telling you about what we've been working on. I have put a question mark up here. Is this the future of organ transplantation? So we don't really know yet. We happen to think that this is a very exciting area, and we see it progressing towards that very quickly. But I guess what I'll kind of discuss today and show you kind of where we came from and where we hope to go. So if we're talking about the future of medical treatments, if you go back about 60 years and you ask someone back then, what is the future of human medicine? The future of human medicine used to look something like this. It was the idea of the bionic human, right? So the $6 Million Man, we've all seen this in movies, and this was science fiction at the time. And over the last several decades, this has become reality. So if you need a total knee replacement, if you need a hip replacement, if you need a pacemaker, people are working on cochlear implants that allow people to hear, and people are also working on retinal implants now. And these treatments have had dramatic improvement in the quality of life of these patients, of course. And the question here is this a general solution for any problem in the body? This is basically the idea that any problem in the body could be treated with a device. And these devices are made of plastics and metals. And what we're realizing now is that there are a lot of organs in the body that can't be adequately treated with a device. So there's a lot of organs in the body that their main function is more of a biochemical function. So we don't know how to take a plastic or a metal and use that to metabolize alcohol, for example. It's a pretty important function. So in our field, the thinking has been transitioning. So these kind of implants will continue to help people. But there's a new idea that has been coming up in the last couple of decades, and it's the idea of regenerative medicine. And so in regenerative medicine, the National Institutes of Health actually picked as their mascot for regenerative medicine Prometheus from Greek mythology. So what gets them excited about Prometheus, about the future of human medicine, is not that he was chained to a rock and had his liver eaten every day, thankfully. It's that his liver regenerated every day. And it's this idea, can we induce or replace organ function in the body? Can we do it in a way that would take your own cells? So, right now the standard of care is to try to get an organ transplant. We definitely need a lot more people to register to be organ transplant donors. So I think that's very important for the short term. But over the long term, what we're looking towards is, can we take cells from a patient, can we put it into a structure that will function like an organ? Can we replace function that way? So then you can think about also, part of the potential here is maybe if you get this type of implant when you're young, maybe it will grow with you and you wouldn't need to get it revised or replaced. So this is really based on the idea that cells outperform devices for a lot of potential applications in the body, and we and others are trying to figure out what are the rules that define that and how could we approach it. So there have been a lot of successes in regenerative medicine in human patients. So these are some synthetic tissues that have been made in the lab that have been used in human patients. So here at the top, this is a synthetic skin graft. So this is they take a piece of collagen. They can take it. Collagen is the main structural component of skin. You can process that. You can make it into a sheet, some kind of this 2D sheet-based material, and you can take a patient's cells and you can sprinkle them over there. You can seed cells on there, and they will grow on that scaffold, and you can use that to implant back in the body. And people are doing this now with cornea replacements, and also this is a urinary bladder here. So you can take a 2D scaffold, you can sew it into a 3D shape. You can see the seams here where they were trying to sew it up into a spherical shape. And these are amazing applications here. And the question that we had is, can we take these sheet-based technologies and can we transition that into something that is more three-dimensional, something like the solid organs, like maybe the liver or the kidney? And so one of the ways that we think about this is sort of a very crude summary of everything going on in our field right now. And we need to think about numbers of cells. So here along the x-axis here, I have, it's a logarithmic scale. So we start at the number 1 all the way on the left here, going up to 1 billion cells. And we are very large. We're thousands of times larger than mice. So we're going to need a lot more cells than what we might use to understand what's going on inside of a mouse. And this is sort of the grand challenge in our field right now. Can we get a billion, maybe 10 billion cells packed together in 3D, and can we keep them all alive in that way? And so why is that a big challenge? If we can grow cells, why can't we just grow a lot of cells and then put them together in 3D? Well, what happens is, you may not realize that's a big challenge. So you go to the deli, you don't realize, if you think about it, chicken doesn't grow as a solid sphere of cells and tissue, right? This is totally artificial. This is not the main structure that we're trying to reproduce. And the reason why that doesn't happen, I have another analogy. It's a bit like baking a loaf of bread. So if you take a big loaf of dough, you put it in the oven, you can turn on the oven, you'll get a crust layer that forms. And the crust layer, chemically, that's oxygenation. So what's happening is the dough is getting oxygen from the atmosphere. Most of that oxygen is taken up on the surrounding outside layer, and all the bread on the inside can't get that oxygen. So it doesn't matter how hot you make the oven, you're not going to get the oxygen deep inside of that big structure. And so that analogy translates directly into working with cells. So here's a scientific experiment that we will do in the lab. So we can make a big loaf, if you will, of cells in a gel. We can pack them all together in 3D. And what happens is if you cut that open afterwards and you look inside what happened, all the cells are only surviving around the perimeter. You still get that crust layer forming where you're only getting cell survival and everything in the center of there is dead. And this is the challenge. So if we want to think about how are we going to make organs for people, this is, it's a very exciting area to think about. And sometimes you wake up at night like, oh, I think maybe we can try it this way. But this is really the grand challenge. Is we're working with stem cell biologists who are taking stem cells. They can make stem cells from adult cells. So the stem cells are the ones that can differentiate into all the different cell lines. So they could make maybe liver tissue. They could make kidney tissue perhaps. And we can grow these in the Petri dish. And we can grow up millions. We can grow up probably billions of cells. But the breakdown happens at this very last step. It's that we can't recreate the architecture of the solid organs. So if you look at the solid organs, the solid part of that is actually a bit of a misnomer. The solid organs are not actually solid at all. The solid organs have this very complicated, interconnected, multi-scale blood vessel network. And this is only one of the blood vessel networks in the liver. So this is the arterial tree. There's also a venous tree. There's also bile ducts. There's also lymphatics. And we can look at this just as a casual observer, and this is a really beautiful structure. If you look at this from the perspective of an engineer, though, this is absolutely terrifying. We have no idea how to make anything like this. We don't know how do we structure it. We are still trying to even understand the anatomy side. So this goes back hundreds of years when we have these images of Leonardo da Vinci drawing the structure of anatomy that he saw. And we're still trying to understand fully what that structure is. So we need to think about how much of this structure do we need to recapitulate to make an organ replacement. And that's something that has been a big challenge in our field for a long time. So one thing that people do to try to grow cells in a big 3D tissue is they can make a sponge-like material. So just like that collagen, you can take a collagen sheet, you can actually process it with gas or maybe salt, something like that, and you can make it into a porous foam like you see up here. There's different ways to do that. It looks just like a sponge you might have in your sink. And you can imagine all these pores, all these holes going through this spongy material. You can put cells in there, and you can also flow fluid through there. You can imagine flowing nutrients and oxygen and trying to get a very high surface area to get enough oxygen in there to keep the cells alive. But what happens over time is the cells will make their own proteins, and that protein comes into this volume that you've put them in. So here along the bottom, you can see over time what happens. The cells are going to start to put this protein matrix in the same volume that you're flowing through, and they will suffocate themselves off. So this is not the best architecture that we would want. And also, a lot is known about how blood works. So blood is very good at clotting, and that's very important as we're walking around and getting injured. And blood also requires certain types of flow. So you have to have very, very organized structures for blood to flow, otherwise it will clot. So when we look at structures like this, we are talking to a transplant surgeon, and you won't be able to get an artery connected directly to a sponge-like material like this. The blood flow kinetics are all wrong, and this is not what we need to do. We need to really think about vasculature. So there are different ways. I won't go into too much detail here. This is some basic biology of how blood vessels naturally form during development when an embryo is growing, and then also blood vessels will form again after an injury. So you have these endothelial cells. These are cells that line all your blood vessels. They keep blood from clotting, and they also control how much oxygen gets into the tissue around them. And this happens at many different scales in the body. So scientists know how to make these endothelial cells make capillaries. So here's a movie of these green cells finding each other in 3D. They're spreading out. They're making actually rudimentary capillary beds in these gels in 3D. So this is very exciting. The question is, can we do this at a larger scale? So can we connect an artery directly to a capillary bed? Well, no, you can't, because the pressures are all wrong. The flow would be wrong. In addition, this type of process takes many days for it to happen. This is a sped up movie over 3 days. And so if you want to imagine putting cells in this big hunk of tissue, maybe 1 billion, maybe 10 billion liver cells, they're not going to wait around for this capillary bed to form. They're all going to die in a couple hours. This is why you have to get a transplant so quickly from a donor into a recipient. The cells just require oxygen. We don't know how to slow that down yet. So what we try to think about is we were trying to figure out what is a way we could do this multi-scale vascularization. So multi-scale, many different sizes of vessels all connected together in a way that resembles close enough native vasculature in the body that we could pump things like blood. And as an engineer, we try to break it down to the simplest possible way that we can and start from there. So we call this the vascular unit cell. So a unit cell is one sort of block, and you can imagine building up these blocks. If you can make that one block, if you can build that up into many larger structures. So those are those three components that I mentioned before. So you have this lumen in green here. This is the hollow part of a vessel where you're going to be pumping nutrients and oxygen and blood. And then we have this vascular wall, those endothelial cells that are holding the blood and preventing it from clotting and keeping all the nutrients going to these cells in this interstitial zone. So one thing that people have been looking at is this idea of 3D printing. And 3D printing, what you can do, this is in bioprinting. So this is, so a lot of you have seen 3D printers now where you're taking a plastic material, you have it, it's sort of like a hot glue gun. You can extrude the plastic, it'll extrude as a liquid, but it'll solidify very quickly again. You can build that up layer by layer to make a 3D structure. And people are looking at this for 3D printing, bioprinting, using cells and this sort of gel material as the ink. And this is very exciting. This is the idea that you could take a bunch of cells, put them in the nozzle, and build up in 3D. Why 3D printing is really exciting from our perspective is that it gives you access to every XYZ position in a volume of interest. So this is very different from other types of manufacturing. Most of manufacturing that we think about is going to be subtractive manufacturing. You think about taking a big block of metal and cutting away everything that you don't want, or maybe you could do things like casting. But in 3D printing, you have access to every different position. The challenge with this type of approach from our perspective has been that cells like the liver cells, these cells that we're very excited to 3D print, they do regenerate in the body, as we said before, but they're actually very fragile cells outside of the body. And we don't have good ways to handle them outside the body. And they don't survive. Maybe you could print 5 or 10 layers of liver cells, but nobody's yet printed the 10,000 layers you would need to be able to make something even the size of an organ, let alone will it function or not. We don't know yet. We're still trying to figure that out. So what we did is we took a step back and we thought about this from the beginning again. So I don't know how many of you saw the Body World exhibit when it was here. So I actually was here in graduate school at Rice University, and I went to go see the Body World exhibit. This is a whole organ vascular cast from that exhibit. And when I saw this, immediately I was thinking, well, this is really what we're trying to make, right? We want to make the blood vessel network. What if we could make this first? What if we could make the blood vessel network first? Maybe something that looks like this, maybe something that functions like this. Then maybe we could put cells around that afterwards, and then try to make this of a material that we could remove it afterwards. And then this would be a temporary scaffold. We could put cells around it to pattern the vascular architecture, and then maybe figure out some way to dissolve it away. So this is the idea that we came up with. We were going to 3D print a lattice structure, something this filament structure put together, and then pour cells and gel all around there. Encase that, solidify the gel, and then start to dissolve away that lattice structure we had 3D printed. And if you could do that, maybe you could make a vessel in the shape of the filament you had initially printed, and then backfill that. Now you can start pumping nutrients and oxygen and blood through there to try to keep cells alive. So this is the approach that we came up with. And how are we going to do this? What is the 3D printing material going to be? Well, it has these two sort of critical parameters that we had to optimize. So number one, it has to be a very strong material because it has to be able to support its structure. But typically when you think of making a very strong material, you're going to cross-link that chemically. And it's that chemical cross-link that's making it stronger, able to be a 3D structure, also directly antagonizes your ability to dissolve it later. So these are antagonistic design criteria. And that whole organ vascular cast I showed before, that's a silicone. You can dissolve that, but any of the caustic chemicals you would use to dissolve that would just kill your cells right away. So it turns out that sugar is actually the best material that we found to do this. And sugar, we're processing it into a glass material. So this is the chemical definition of what glass is. So it's this unordered liquidy material. And this is not, there's no silica here. So sugar glass, if you've ever seen a movie when somebody breaks a bottle over somebody's head or somebody jumps through a window, that's actually made of sugar glass. So it's a very rigid material. It can hold a form, but we're using sugars that are already found in your bloodstream. So then we knew it would be biocompatible. So this solves both of these problems for us. So we're able to process the sugar glass, and we can do it in the lab, and we can pull these filaments. You can read text through it. We've made it very optically clear. We've reinforced it so it can hold its own weight very strongly. And I'll just jump to the video here. This is the process, this is how it works. So we have this single filament of sugar, and I just poured a gel. You can think of it like Jell-O, where you're just pouring a liquid, it'll solidify. So you can imagine eventually we'll put cells there. That, and this is a sped up movie, that gel has solidified. Now we're pouring salt water all over there, and the sugar has dissolved in place. We're gonna push with a pipette a little bit of fluid, and all the dissolved sugar is gonna flow right out there. And that's how you can make one single filament, one single vessel now in one of these biocompatible gels. Now, one vessel is not enough to make an organ. So how are we going to scale this up? How are we going to make it more complicated? And that's where we came to 3D printing. So this is the RepRap open source 3D printer project. It's an online movement that has really revolutionized what a lot of people think about 3D printing. This came from a researcher in England, and he had the idea of a mostly 3D printable 3D printer. So he wanted, you can see all the white parts here that structurally make the 3D printer. They are themselves 3D printable. So he envisioned it sort of like a 3D Xerox machine where it could keep making copies of itself, and people could modify it. You can print your own upgrades for this printer. So it was really exciting when we heard about it, and it's open source. So a lot of times you try to talk to commercial companies, and they don't really want to work with you because they don't really see the business model yet, and it's very hard to do science that way. So what we did when we came to this group, and we started in our hackerspace trying to figure this out, we started emailing people all around the world in this project, and they were really willing to help us to figure out how to modify this. So open source is great. You can legally download all the designs, but as you may know from the Linux operating system, so Linux is free, but your time, only if your time is worthless. So that's kind of what people have thought about this open source, is that it's so hard to get, oh, sorry. Okay, maybe I just don't need that. It's so hard to get enough documentation to understand what's going on that it'll take you a while. But if you do figure that out, then you can do some really amazing stuff. So this is what we did. We modified this open source 3D printer that we 3D printed, and we are able to extrude this sugar glass here. So you can see we're building up this wall made of molten sugar. So it's coming out as a liquid, but then we blow air on it and we freeze it again. And this allows us to print these single filaments of sugar, which we can then pour a gel around, solidify that, and then remove it away to make vessel networks. So what we wanted to do is try to figure out how do we make these multi-scale structures? How are we going to make many different diameters of these vessels? And initially we thought we'd need different nozzle sizes for every different vessel that we wanted. And what we realized is because this is a glass, we could use the laws of glass fiber drawing. So this is the same way that wire is made, the same way that fiber optic cable is made. You can just heat it up and you can pull whatever diameter that you want. And so with that technology, we're able now to make these very intricate structures. We can make multi-layer structures. This is a multi-scale vasculature you can see up here, where you have a 1 millimeter diameter filament along the top, and you have these smaller branches coming off. They're somewhat reminiscent of structures you might see in the body. And we're looking at ways to optimize this a lot more. So I guess I'll just jump over here. There's a lot of different types of materials that people are using for cells. So liver cells don't grow in the same type of gel that kidney cells do. They're not optimized the same way. So we wanted to make sure we could use any type of gel material that scientists currently like to use. And so this is just some evidence showing that we could mold these vessels in any type of matrix, any type of gel material. And if you can do any type of gel material, you can also do any type of gel concentration. So you can make these gels very dense, such that they can mechanically, robustly support blood flow here. So this is human blood that we're pumping in through here. It's going across these vessels, and the gel is mechanically intact, even though it was molded from these filaments of sugar. So there's one vessel there. You can do this across many much larger vessels. We're pumping in blood at arterial flow pressures here, and the gel survives that very well. We get these very interesting flow patterns that are very similar to how blood flows in the body, where you actually have blood flowing in a spiral down cylinders in the body. All the blood vessels are cylinders, and we're able to make that with these sugar filament inverse molding techniques. So then we do the experiment again. What if we go back to that slice of bread analogy? How do we see what happens on the inside? And what we find actually, so in contrast to when we have just a big block of gel and there's nothing in the center and we just have cells, they all die. What you see now, if we take one of these gels that we put these vessels in and we're pumping nutrients and oxygen in there, we can keep a lot more cells alive, and that's as a function of the vessel architecture that we had 3D printed. So here we're starting to build up the rules that we need to follow to be able to make these more complicated tissues. So here's a zoom in. Every point of light that you see here is one single cell, and each of these vessels is about 800 microns in diameter. A micron is a thousandth of a millimeter. So this is about 0.8 millimeters in diameter. And you can still see we have a lot of dead cells around here, but we're starting to understand now what are the architectures we need to use to be able to keep cells alive. So this cell line was not one that was really interesting from a therapeutic perspective. So next we want to look at these liver cells. And what happens is these liver cells, we were able to show that when we took liver cells, we put them in this gel, they performed better in the gels that had these vascular channels in there. And just like that other cell line, this is another way of looking at it with a live dead stain. We can show colorimetrically what cells are alive and what cells are dead. And so this is not ideal. Again, we still have these dead zones, but we know that we're 3D printing areas that become alive. And so what this is going to allow us to do, we're using now the power of and the maturity of computer systems. We can structurally analyze the tissues that we're making. We can then make a computer model for how the cells will survive as a function of the architecture, and then redesign the architecture on the computer and simulate when cells will be alive and when they won't, and then use that to feed back to the 3D printer. So that's what we're trying to do now, is take these designs, create the tissue, mathematically understand what's happening, make a computer model of that, and then use that to refine. And this is the typical engineering you'd want to do, where you're going to iterate and you're going to cycle back and you're going to improve. So some of the new things that we're looking towards as we look towards the future. So this is a very interesting study that was published recently, where some researchers are looking at how capillaries form into those larger vessels over time. So these are 2D drawings of some images people took to analyze. What you see here, all these single dots are individual cells, and they're in this pink area, which is all the vessel structure, all the capillaries. And these capillaries fuse over time and become these larger vessels. So what we're thinking about is now we have this 3D image of how cells are growing. This is done by imaging, and that image data is actually 3D data. So we're trying to take that 3D data, invert that, extract that vascular architecture, turn that into 3D printing instructions, and then try to print out structures that will let us analyze more how these capillary beds form into larger vessels. And then as we look towards the future, sort of longer term, we're really excited about this idea of whole organ vasculature. So here's one way that we're starting to think about now and trying to get proof of concept, where you could take that angiography I showed before, that micro CT, that CAT scanning of an organ, where you're analyzing all the vascular architecture. You can extract that as an image, make that into a 3D model that maybe you could 3D print. So we could do maybe a native vasculature there. Maybe we could modify it. We could create a new vessel architecture totally from scratch on the computer, and then try to 3D print that. So I also just wanted to emphasize that I had such a good time in this research working with people all around the world, working with people in the open source community. We decided to try to formalize this interaction. And so one thing that we did last summer at Rice is we launched this Advanced Manufacturing Research Institute, where we were trying to formalize the interactions between people in the hackerspace, the makerspace, the maker movement. There's a similar maker-type movement where people are tinkering as hobbyists with biology, so DIY bio, people working with bacteria, and people in the lab. So I thought one thing I noticed is a lot of people didn't have a lot of respect for the maker movement, a long time ago because they thought, well, these people aren't really taking things seriously and they don't really have a lot of skills. And I just didn't believe that because I saw what they were capable of. And in fact, if you think about it, really, the reason that they weren't taking things seriously, they were doing it as a hobby, right? So obviously it wasn't their job. So the idea here was that they're such capable people, if we can just show them a little bit more about the scientific method, then maybe they can actually be interested in going into science more. So we had these fellowships last summer where people came to the lab. They worked with us in the lab. We provided sponsorship. We provided scientific mentorship. And you can find out about this more online, but basically what these fellows came in to do is they were modifying these different materials to use them for bioprinting. So you can use a projector, you can use inkjet to try to do 3D printing of tissues, you can use syringe-based materials to extrude that bioprinting that I was showing before, and then some of our latest stuff, we're trying to use lasers to come in and melt the sugar to make a much more complicated vascular architecture. So what they were trying to do is the engineering design phase, trying to teach them engineering, and then maybe this summer they'll be able to do more with science. So we had to define and design and develop their hardware, and then we were slowly teaching them how to start to measure things that they were doing in the lab. So we're hoping to continue that this summer, and I guess you can check that out more online. So with that, I'll just leave you with an image here of our latest approach here. This laser sintering is much better than melt extrusion. We're trying to do that to create whole organ vasculature. So thank you. I'll be happy to take any questions. Yeah.
What's the working temperature of your printer?
For something like this?
Yeah.
So it really depends on the material that you want to print. So if you want to do something like nylon, for example, you need to keep it around 60 Celsius, something like that. If you want to do something, some people are doing 3D printed metal now. So if you want to do metal, you have to get metal powder. You have to do it under nitrogen gas because it's extremely reactive, and you have to heat it up to several hundred degrees Celsius. But you can do that too. So it really depends on what the material is that you want to print.
What about the sugar glass?
So the sugar glass, you can do that about 100 Celsius, so about boiling temperature, and it's great. It comes out. It's very strange because it goes through this glass transition where it's liquidy and rubbery, and then it very quickly solidifies. So we actually learned a lot of. We started with a recipe from the confectionery industry. We really did, and then we just kept modifying it to make it more rigorously scientific and a bit more stiff so that we could use it. Yeah.
How does like stem cell transplantation? I mean, like our company has a stem cell arena using the stem cells to regenerate within the body. How do you combine this kind of a technology for you?
Yeah, I think. Yeah, I think.
Take that.
Yeah. So the question is about people working with stem cells. How could they utilize technology like this? So I think that it's a very good. We've tried to, from an engineering perspective, you want to separate the challenge into as small a part as you can. So we were focused exclusively on architecture, and the cell source I think is a very interesting question, and it's not something we have a lot of expertise in. But there are stem cell biologists all around the world that are doing these amazing things with single cells and with cell colonies and getting primary stem cells from the body or from the placenta, like you said. So I think that we are sort of agnostic to what type of cells you could use. We're mostly just focused on recapitulating the architecture and then making a platform that people could use for all different kinds of applications. So I think that because you have 3D printing, you have absolute reproducibility of the structures you're making from day to day. You're not going to have this sort of one-off print. You can do hundreds or potentially, if you get a little bit more people, you could do maybe 1,000 of these in one day. And we're getting to where we can do rigorous scientific experiments now with vascular tissue that we couldn't do before because we just didn't have the 3D printing technologies. Yeah.
Once you have the sugar glass structure built and then it's encased in that gel, does it break down naturally, or do you have to sort of kickstart it?
Yeah, it dissolves very quickly actually. So there's a very subtle trick that we use where if you can imagine any kind of sugar, if you put it in water, it's going to immediately dissolve. So we're actually using a candy coating on the sugar glass. So we spray it with this biocompatible polymer, and it keeps it from dissolving right when we pour the gel, but it's still a little bit porous. It has very small pores in it. It's able to suck water out of the surrounding gel. Then it dissolves in place, and then we can pump it out again. So we know that even though sugar is biocompatible at very high concentrations, osmotic pressure would just kill cells if the sugar dissolved through the gel where the cells are. So we really had to figure out a way to dissolve it in place so that we could just flow it out of the network. Then it would be diluted in the bulk of the solution, the bulk of the salt water, and not kill the cells. So, and now we're trying to look at other sugars that are still very good structurally, but maybe cells can't even metabolize them, so we think they won't have any effects on cell physiology. So usually about 30 minutes after making the gel, we can dissolve all the sugar and remove it. So the cells are only exposed for about 30 minutes or so, and we've seen some pretty good results so far. So, yeah.
What do we know about how nature makes these amazing vascular structures, and can that inform, or does that inform how you guys choose to do the printing in terms of the process?
Yeah, absolutely. We have this debate a lot, is how much of the vascular architecture do we need? So we're not looking right now at 3D printing the capillaries. So a lot of people had told me, oh, you really got to do the capillaries because that's where all the oxygenation happens. But I believe that since the cells are so good at making capillaries, and we also know that if you have cells in a capillary bed, they will remodel it over time. They will prune different capillaries they have. They'll take them away and they'll grow new ones based on local needs that they're sensing from cells in the interstitial space. So what I think that we'll have to do is just get close to the capillary level and then let the cells make their own capillaries. So actually I have you can see it right here. We have some evidence that that can work. So this is some of these cells in red here that are these endothelial cells that can make capillaries, and we're looking through one of these gels. So I'm going to show you a movie, and that movie is, imagine taking serial slices through here. You're going to be looking down this channel. There's a 200 micron channel here. It's about the diameter of human hair. And what these red cells do is we are finding now they're able to make new capillaries, which you see right there, sprouting off of the vessels that we're making. So I think that we won't have to make the capillary bed. We'll just have to make something close to that, let them make their own capillaries. And maybe we can give enough nutrients and oxygen that the cells will survive. So you can see that better here. There's a filament that we had molded around that has that hollow bit in the center we can flow, and these red cells are making new capillaries coming off of there. So we're still trying to understand this better. The challenges are pretty tough to even image what's going on in there, but I think it's something we'll be looking at for a while. Oh, one more question?
Sure. So there's starting to be some commercial sugar 3D printers.
Mhm.
Have you been talking to the commercial industry as well, or have you been relying mostly on open source makerspace type of technologies?
Yeah, there was this really elegant sugar printer that came out that I think it was a husband and wife team that they were making. They would take sugar powder and come in and spray droplets of binder to bind the particles together. And they're made these really intricate structures. They were recently acquired by, I can't remember if it's 3D Systems or maybe Stratasys, yeah. So we haven't been talking to them because we actually need the sugar to be in this glassy state. We need it to be transparent, and we need it to be able to, the reason we need it transparent, we have some gel material that we can put cells in that we can cross them with light, so we can just shine white light on it. It induces a chemical reaction that will gel, that will solidify the gel. So we need to keep it transparent, but I think a lot of the structures that they made are very intricate and